Hello everyone, this is for B.Sc course 6th semester paper 8 biochemistry. In this session, I am going to tell you about the fatty acid metabolism. Before understanding the fatty acid metabolism, we'll see what are lipids, fatty acids, and the different steps involved in the fatty acid metabolism. See, in this figure what you are seeing are all lipids. Why this is very important, the metabolism of fatty acids is, see these triacylglycerols and the adipose tissue fats are the major source of fatty acids. See, oils and fats what we take in our diets are nothing but the lipids. Okay. Now, the cells uses maximum amount of energy from these oils and fats. They are an efficient source of energy and they provide the energy more than the double when compared to carbohydrates. That is around 38 kilojoules per mole on oxidation, or it is 9 kilocalories per mole upon oxidation. Now, what are these lipids? See, lipids are the macrobiomolecules. They are soluble in non-polar solvents and insoluble in water. Some of the examples for these are fatty acids, wax, fat-soluble vitamins such as A, D, E and K, etc. Okay. See, the function of these lipids, the main function of these lipids include storing of energy. Okay. And they act as a structural component of cell membranes. Coming to the chemistry of these lipids, they are chemically fatty acids and fatty acid derivatives. Okay. And they are divided into 8 different categories as shown here. Right? Now, we know very well carboxylic acids have the general formula RCOOH. Then what is this fatty acid? If this R here is a long chain, then they are known as fatty acids. This long chain may be saturated or unsaturated. Depending on that, we have saturated fatty acids and unsaturated fatty acids. Saturated fatty acids example are butyric acid, lauric acid, myristic acid, palmitic acid, stearic acid. Unsaturated fatty acids are oleic acid, linoleic acid and then linolenic acid. So remember, fatty acids will have got a long chain. R is a very long chain. So this is a general formula for fatty acids, how we represent. And this is the formula specifically palmitic acid. That is CH3, CH2 13 times, CH2, COOH. Okay. Now, so this is the structure of or how you can represent a fatty acid. COO minus a carboxyl end. Okay. So this is a hydrocarbon chain. Right. Now, coming to fatty acid metabolism. As I told, fatty acid metabolism plays an important role because most of our energy is derived from these fatty acids, nothing but oils and fats. Where do this metabolism occurs? In liver cells. Okay. And fatty acid metabolism occurs in these steps. Number 1, activation of fatty acid. Number 2, transport of fatty acid, that is transport of activated fatty acid, and then the beta oxidation of fatty acids. And remember, the fatty acid metabolism occurs in liver. Okay. Now, see, this fatty acids, that is inactive fatty acid which is in cytoplasm, is converted into active fatty acyl CoA because this just fatty acid cannot enter into the mitochondrial matrix. Okay. So it has to be activated, and this activation occurs in 2 steps with the help of an enzyme thiokinase. So this is the activation of fatty acid. Say RCH2CH2COOH. or COO minus is an ionic form, is a fatty acid. So with the help of an enzyme thiokinase, this group is getting activated here. COA, coenzyme A getting attached here. So this step is called as the activation of fatty acid, which takes place in the cytoplasm of the cell. Now we have got activated fatty acid. It has to be transported into the mitochondrial matrix across the mitochondrial membrane. How does it take place? It requires a transport molecule known as carnitine. So, carnitine plays a very important role in this fatty acid metabolism because with the help of this carnitine, our activated fatty acyl molecule gets entered into the mitochondrial matrix. See here, we have activated fatty acid combining with this carnitine. With the help of a carrier protein, it is going inside the mitochondrial matrix. Okay, so the role of carnitine is to transport activated fatty acid into the mitochondrial matrix through this reaction with the help of carnitine acyltransferase 1 and then carnitine acyltransferase 2. So they'll ask you for 2 marks. What is the role of carnitine in beta oxidation pathway or not in, if not in fatty acid metabolism? Okay. So now we have understood 2 steps. One is activation of fatty acid. Number 2 is transport of this activated fatty acid into the mitochondrial matrix. Now the process of beta oxidation starts. So let me tell you briefly. See, this beta oxidation pathway was illustrated by F. Noob, a German biochemist, and this takes place in 4 steps. So what happens in these 4 steps is this activated fatty acyl CoA molecule through these 4 steps in each cycle. One cycle of beta oxidation means 4 steps. In these 4 steps, it goes on eliminating 2 carbon atoms in the form of acetyl CoA. So in every cycle of beta oxidation, the long chain of this fatty acyl CoA gets shortened by 2 carbon atoms in the form of acetyl CoA, where this acetyl CoA then enters into CTA cycle and the remaining fatty acyl CoA again enters into beta oxidation pathway. In every cycle, the long chain is shortened by 2 carbon atoms. How? In the form of acetyl CoA. Let us understand the reactions now. So these are the 4 reactions of beta oxidation. So which are those reactions? First one is dehydrogenation, hydration, dehydrogenation and the cleavage. So which are the 4 reactions? Dehydrogenation, hydration, dehydrogenation and the cleavage. Now coming to the reactions. So this is fatty acyl CoA. In the first step, it undergoes dehydrogenation with the help of an enzyme acyl CoA dehydrogenase. See, all these are the 4 enzymes. Okay, acyl CoA dehydrogenase, enoyl CoA hydratase, beta hydroxy acyl CoA dehydrogenase, and then beta ketothiolase are the enzymes involved in the beta oxidation step. See, in the first step, you have fatty acyl CoA here. See, the carbon atom next to this functional group is designated as alpha carbon and this is beta carbon. The oxidation or the dehydrogenation starts from this carbon atom, hence the name beta oxidation. See, with the help of an enzyme acyl CoA dehydrogenase, it begins with FAD. This FAD accepts this hydrogen atom and undergoes reduction to FADH2. So in this step, a molecule of FADH2 is produced and you get a double bond in between here. So in this dehydrogenation, with the help of acyl CoA dehydrogenase, a molecule of FADH3 is produced producing trans, you can see here hydrogen atoms are trans to each other, trans-enoyl CoA. In the next step, addition of water, hydratase. With the help of an enzyme enoyl CoA hydratase, you have a double bond here. Open up this double bond, addition of H and then OH. In the next step, so this is beta hydroxyacyl CoA 1, 2. It is also known as 3-hydroxyacyl CoA or beta hydroxyacyl CoA. In the next step, dehydrogenation takes place. We have a secondary OH group here. CHOH is a secondary OH group. Upon oxidation, you end up with a keto group here with the help of an enzyme beta hydroxyacyl CoA dehydrogenase. Now, this gets cleaved here. This gets cleaved. See, these two carbon atoms comes out in the form of acetyl CoA. You have left out with the fatty acyl CoA, which again enters into beta oxidation, and this acetyl CoA enters into Krebs cycle. This acetyl CoA enters into Krebs cycle. Coming to the reactions, see, in the first step, you have fatty acyl CoA, dehydrogenation or oxidation taking place. Okay. Now, so here you see reaction starting at the beta carbon atom. We have got trans-enoyl CoA. In the next step, addition of water, that is hydration. So in this double bond you open up, add H and then OH. Okay, you should learn to write the reaction. That's why I put the reactions in the slide. Along with the flow chart, please learn to write the reaction stepwise. Now, coming step 3, again you have beta hydroxyacyl CoA with the help of an enzyme beta hydroxyacyl CoA dehydrogenase. You see you have a secondary OH group here, CHOH getting converted to a keto group here. Okay, so this is beta ketoacyl CoA. In the next step, there is a cleavage here with the help of an enzyme acyl transferase or thiolase. See, the two carbon atoms from the carboxylic end gets cleaved in the last step in the every cycle of beta oxidation, producing acetyl CoA molecule. So now you have learned one complete cycle of beta oxidation through 4 steps. What are the products obtained? One is acetyl CoA, fatty acyl CoA. So the long chain of fatty acid is now shortened by 2 carbon atoms. And where does this 2 carbon compound has gone? What you have got is acetyl CoA, which enters into Krebs cycle, and this fatty acyl CoA again enters into beta oxidation. So every time the ending 2 carbon atoms from the end of carboxylic end getting cleaved in the form of acetyl CoA. Okay. So these are the 4 steps involved in the beta oxidation process. Now, so the same thing which is again shown in the flow chart. Okay. So learn all the 4 reactions of beta oxidation. See here, acyl, you have got this compound where 2 carbon atoms are shorter. Now, after knowing the beta oxidation pathway, so now you know very well, fatty acid undergoes activation. Activated fatty acyl CoA enters into the mitochondrial matrix with the help of carnitine. Once it is inside the mitochondrial matrix, the reactions of beta oxidation sets in through 4 steps. Okay, and in every step, 2 carbon atoms gets reduced, or the long chain is shortened by 2 carbon atoms forming acetyl CoA and then again forming fatty acyl CoA. Now, particularly let us take the example of palmitic acid. See, the formula of palmitic acid is CH3, CH213, CH2COOH. Count together. So what you have? C, this is the 13, 14, 15, 16. Okay. So it is C16 compound. Palmitic acid is a C16 compound. So you have 16 carbon atoms in palmitic acid. So you will be asked to write the energetics of palmitic acid when it undergoes beta oxidation. So, before that, in beta oxidation pathway, you have one FADH2 released and then one NADH release. See, every cycle of beta oxidation produces one FADH2 and then one NADH. These formed enters into ETC. They enter into ETC. One NADH is equivalent to 3 ATP. One FADH2 is equal to 2 ATP. So remember, when beta oxidation takes place through these 4 steps, you have totally 5 ATPs released per cycle of beta oxidation. Now, coming to this palmitic acid, when palmitoyl undergoes activation, what you get is palmitoyl-CoA. You have 16 carbon atoms. Okay. See, when even number of carbon atoms containing fatty acid undergoes beta oxidation pathway, 2 carbon atoms at the end gets on eliminating. So now, this is a 16 carbon palmitoyl-CoA contains 16 carbon atoms. So when it breaks through beta oxidation, what you get is 8 acetyl-CoA. How many acetyl-CoA you get? 8 acetyl-CoA. Because how many carbon atoms are there in palmitoyl-CoA? There are 16 carbon atoms. So when it undergoes beta oxidation, the product what you are going to get is 8 acetyl-CoA molecules. Now, in how many cycles is very important. It gives 8 acetyl-CoA molecules in 7 cycles of beta oxidation. So now we will see how this 8 acetyl-CoA enters into 7 cycles of beta oxidation. Please remember, one cycle of beta oxidation will give you 5 ATP molecules. So, this 8 acetyl-CoA then enters into Krebs cycle. How many rounds? 8 rounds. Now, so this is palmitic acid. This is palmitic acid, a 16 carbon compound. Some of you will get it out how it is 16. You have to count this carbon also. Now, see, it undergoes 7 cycles of beta oxidation. So many of you will get confused. Why not 8 cycle? It can't be 8 cycle because once it undergoes 7 cycles, what you get, the last one remaining is acetyl-CoA only, which contains 2 carbon atoms. So it gives you 8 acetyl-CoA in 7 cycles of beta oxidation. Now, in every cycle of one beta oxidation, it gives you one FADH2 and one NADH. One FADH2 and one NADH. Remember, this acetyl-CoA enters into Krebs cycle. When one acetyl-CoA enters into Krebs cycle, the number of ATP released is 12. We have learned this. When one acetyl-CoA enters into Krebs cycle, the number of ATP released is 12. From 1 FADH2, 2 ATP. From 1 NADH, 3 ATP. So, one cycle of beta oxidation will give you 17 ATP. Now, so totally what you have. So, this palmitic acid will give you 35 ATPs. Okay, 35 ATP released from one molecule of palmitic acid when it undergoes beta oxidation. Here you have 8 acetyl-CoA produced. Those 8 acetyl-CoA enters into 8 round of Krebs cycle. Now, there the number of ATP released is 96. How it is 96? I have shown here. One acetyl-CoA entering into Krebs cycle will give you 12 ATP. 12 into 8. How much you get? 96. Now, so the total number of ATP released from one molecule of palmitic acid is through beta oxidation 35. From 8 acetyl-CoA molecules 96. 131 ATP. And 2 are utilized in the activation process. So you have to subtract this. So 131 minus 2, how much ATP you will get? 129 ATP molecules. So what we have learned so far is, what are these oils and fats, and the importance of fatty acid metabolism, and also fatty acid metabolism is nothing but beta oxidation process. It's nothing but beta oxidation process. Here you should know how this fatty acid undergoes activation and how this activated fatty acid molecule enters into the mitochondrial matrix with the help of carnitine, a transport enzyme, and the 4 steps involved in the beta oxidation process, and the energetics of palmitic acid when it undergoes complete degradation through beta oxidation process, and whatever the acetyl-CoA released from this enters into Krebs cycle. The number of ATP released from one palmitic acid undergoes complete degradation is 129 ATPs. Thank you.